so I just wanted to go over some examples today, guys, of our DNA going to RNA, going to a protein, and give you all the different scenarios that we could do given those different variables. So here's our codon chart. Remember, codon charts are always going to be mRNA. So these ones, you always need the mRNA to put our triplet codes in there. Same thing with this one, all right? It's still a codon chart, just a different way. I need you guys to make sure that you know how to use both of them. So I'm just gonna have a very long sequence of DNA. I'm gonna give you an easy example, and then we will get more difficult as I go along. So here's our DNA. Let's just do A, TTA, CGC, ATG. Okay? Now, if we wanted to take that and make it into mRNA, we could. A would go to U, A, U, G, C, G. A goes to U, T goes to A, and G goes to C. Hopefully that's pretty easy for you. Now, I'm going to write the protein down here just to show you guys what goes on later, and I'll fill in this little blank space here. Now, in order to get our protein, you guys, and they're on your canvas, so you can go to Canvas, but we're going to use your codon charts. Now, I have mine right here. I have the circular one. I have this one. Whatever one you like to use is fine, but make sure you can use both in case one of them is on the keystone and the other one is not. So they're only going to give you one of them on there. Now, if we look at the first one, U, and if you guys have your chart on there, you know, you can follow along, but U goes to phenylalanine. So we'll just do PHE. AAU goes to asparagine, so ASP. And you don't have to know all these amino acids, guys. You'll have a chart that says them all. GCG is our next one. GCG. And notice I'm always using the red. I'm always using the mRNA for this. And then UAC is tyrosine. Okay. So here is our protein, our nice sequence. Now, again, we're always using the mRNA in our codon charts. Always, always, always the mRNA. When we are bringing in these amino acids, these proteins, okay, whether it's phenylalanine, tyrosine, so when they are coming in, they are actually attached to something that looks almost like a clover. Just like that. And on the other side of this, I'm not going to try to get artsy on us here. Let me just draw a two-dimensional figure instead of trying to get 3D. So on the other side of this, carrying the phenylalanine over, the blue is our tRNA. So that is called transfer RNA, and that's going to transfer our amino acid over. If we wanted to see what was in these three positions, U would go to A. If we want to do the next one, good enough. A would go to U, A, because this is tRNA. This one, I'm not going to draw all the clovers here, but it would be C, G, C. This one, A, U, G. Okay? So that is the tRNA. So we can use the mRNA to get the tRNA, but we always use the mRNA right here to put into those codon charts and get our protein out of it. All right, so that's your first example. Okay, so I made another DNA sequence for a second one, guys. This one, it's going to be a little different. So here is the question. The question is, for this example, give me the amino acid sequence. Give me the amino acid sequence for the complementary DNA. Give me the amino acid sequence for the complementary DNA. This is not the complementary DNA. That is our first step. We have to go to the complementary DNA before we do anything. So, complementary DNA. All right? So, T is going to go to A is going to go to T, C is going to go to G. C is going to go to G, C, A goes to T, C, T, C, G, C, A, and then A, T, A. Okay? So we went to the complement first because it's asking for the amino acid sequence for our complementary DNA. Now we can take that and go to mRNA. So A is going to go to U, T is going to go to A, and G is going to go to C. And if you notice, it's the same as the original DNA, except we're swapping out the T's for U's. So you can do it either way, whatever. You'll get the same answer. So CGA, GAG, CGU, and UAU. Now, I can go to tRNA. I know we don't need to. We do not need to go to tRNA, but we can if we want to. The tRNA, and why I'm doing this is just so you guys can see that the tRNA, U goes to A goes to U, C goes to G. tRNA is actually going to be the same as the complementary DNA, except for instead of the T's, there's going to be some U's in there. All right. Now we can go to our amino acid sequence or our protein, whatever you want to call it. So once again, using your codon chart, make sure you're using the red though. So our first protein is UAC, which is tyrosine. So TYR, that's going to be attached to GCU, which is alanine. CUC, leucine. GCA, that's also alanine. And then AUA, that's isoleucine. Okay? So there is our nice protein sequence there. Okay? And you guys can see the wording was different. The last one, we just used the DNA. For this one, we use the complement DNA. So the complementary. So we use the other strand as our DNA. Okay? So just another example I wanted to go over with you guys. I also wanted to show you, if you want to, let's say you get tRNA. And let's say the problem asks you to go to DNA. That's intimidating, isn't it? All you're doing, guys, is working backwards. So you'd have to go to mRNA, which is A goes to U, C goes to G, A goes to U, C goes to G, G goes to C, and U goes to A. Then we can go to our DNA. If it doesn't ask us for the complement, we only have to go to one DNA. U would go to A, C, A. And then G would go to C, C would go to G, and A would go to T, not U, because it's DNA. All right, so we can work backwards too, depending on what the problem gives us. But make sure, guys, you know how to use both of these charts. I got a few practice problems for you to do on the worksheet, and that's pretty much it. Okay, so not too long of a video. So try out the practice problems. I think the practice problems don't use the complement, so it's just going from DNA to mRNA to the amino acid. So pretty simple. But later on down the road, we might use the complement for it, okay? So that's pretty much it for today, guys. So work on the worksheet. Have a good rest of your day.